Well, hey there. Good evening, and it's good to see you again. Here I am with my second installment of Chapter 4 of Drug, the Science and Culture Behind Psychotropic Drugs. In the first part of this chapter, I presented a few sections that focused on the rise of pharmaceutical industry in USA, the rise of Eli Lilly and Company, which presented to the world some of the very life-saving meds and established the local pharmacology manufacturing as a global competitor and became a titan in the field. In this second part, I will continue to talk about the rise of USA pharma industry, especially Eli Lilly and Co. Involvement in the development of psychotropics and how US psychiatry pioneered this field. Let's remember the book is about the culture and science behind psychotropic drugs. To talk about any drug or psychotropic drug, the author investigates the circumstances that led to them and yielded them, how they were tested, how they act, and how they act the way they do. By doing so, the author investigates the disease itself, what causes it, physiological imbalances, stressors, exhausting certain physiological reserves, genetics, etc. That is how, in the section of psychology of depression, which is the first I am talking about, I'm going to talk about in this part, the author talks about how in the 1950s uh, uh, psychotropic drugs were initiated, and the modern psychiatry uh, has started, and how certain diseases are now not referred to the way they were referred to as melancholia, but uh, uh, such as depressive disorders, and we know now about psychotropic drugs, anxiolytics, antipsychotics, antidepressants. Psychiatrists started prescribing psychotropic meds to both patients and outpatients. Depression and other mood disorders became clinical diagnoses. Um, Links have been established among depression and other mental and physical disorders. The impact of psychiatric disorders on patient lives and lives of the people around them were also established. The next section is titled "The Nazis' Largesse." This section talks about how the first antidepressant came to life. Warfare. You do not believe it. It's Nazis. Unbelievable, but it's true. That is how I believe the title denotes both sarcasm and irony. Sarcasm, largesse, means generosity and refers to all the warfare reserves that were left behind, that were not meant to promote health, yet they ironically did. After they had been defeated, the Nazis were forced to leave behind a lot of their artillery, ammunition, and science experiment they were working on. As World War II was about to end, Hitler pulled another stunt. He raided Europe, specifically London, using lethal V-1 and V-2 rockets. The attacks claimed lives of thousands of people and caused substantial infrastructural damages. It was believed that Hitler was on his way to industrializing the most deadly rocket warfare has ever conceived, a rocket that could have ended it all in his favor. Uh, US was also believed to be his next target. When the Allies won, they led their hands on the Nazis' war reserves. The USA laid hand on the secret base where experimental weapons were developed and on the engineers that were behind those experiments. Werner von Braun, the genius Nazi scientist, behind the secret project weapon was one of the captured scientists. The Allies had found a large stock of sea stuff in the rocket base. It is a chemical substance that contained highly reactive and unstable hydrogen uh, used as a propellant for V2 rockets. To bribe the captured Nazi Van uh, Werner von Braun into cooperation and giving secrets about the lethal weapon he was working on, together with secret reserves for hydrazine, CIA decided to get involved in the pharmaceutical industry to raise funds for warfare industry through pharmacology. They involved the local Eli Lilly and other local rising pharmaceutical companies. Why take talking all about this? Why talking about all this? Because this is exactly what led to the manufacturing the first antidepressants. How? This is what the author talks about under the headline "The First Antidepressants." What to make of all uh, this recovered Nazi hydrazine reserves and the highly unstable substance? It can be used in many chemical reactions. Hydrazines or derivative of hydrazines were first discovered in 19th century, specifically in 1874 by the chemist Emil Fischer, a student of Adolf von Baer. He synthesized phenylhydrazine. Hans Meyer and Joseph Marley, doctoral students, synthesized isonicotinyl hydrazine in 1912. It was resynthesized in 1950s. USA was trying to find a more effective treatment for tuberculosis than streptomycin, to which the microbacterium had grown a resistance. They ran many tests and found chemically related structures to the compound thiocyanate have been somewhat efficient in treating infected mice. The synthesized chemical uh, the they synthesized many chemically related structures and analogs. In 1951, Squibb Institute synthesized isonicotinyl aldehyde, thiocyanate isonicotinyl hydrazine was used as intermediate. And isonicotinyl hydrazine that was used as an intermediate was found to be far more potent as anti-tubercular than the final product isonicotinyl aldehyde thiocyanate. Hoffman-LaRoche scientists used nicotinamide, a vitamin B group, combined with thiocyanate, searching for the right anti-tubercular. They also used isonicotinyl hydrazine as intermediate. And guess what? They found that it was more potent as anti-tubercular than the final product. And isonicotinyl propylhydrazine, another analog that was synthesized, was also found to be a good anti-tubercular. They decided to harness the anti-tubercular potential of isonicotinyl hydrazine. Now they tossed away the other chemicals and they decided to focus on this one. Tested on mice, it showed a great efficacy and treated the infected mice. They rebaptized it, they renamed it, the product, as isoniazid and iproniazid, tested it on infected patients. Its efficacy as anti-tubercular was proven. Was proven. An experiment um, that was run at Seaview Hospital for treating patients in New York showed the patients improved physically and also exhibited an extreme lightness of mood. This was more noticeable with iproniazid. They are no longer depressed and they felt good about themselves and they jived and all that. Iproniazid was marketed as Marcelid by Hoffman LaRoche. In chapter 3, if you remember, it was observed that some antipsychotic, which we talked about in the first chapter because the author talks about them, and because it's also introduced to us how antipsychotics were burdened and how they were searched. And just like what we are going to see, they were an accidental find. So, in the third chapter, reserpine, the antipsychotic, depressed patient motor activity and made them lethargic and sometimes even catatonic and had an overall depressive effect. Nathan Klein, a famous American psychiatrist, pioneered the psychotropic modality treatment for mental health disorders. He reported that antipsychotics need an adjunct therapy. He was the first one who used those antipsychotics and noticed all those side effects and said in a conference he said that he needed an adjunct therapy. This therapy should be also drug based and it should act as a psychic energizer to counteract the depressive side effects of antipsychotics such as reserpine. In 1956, Nathan Klein delivered a lecture at former Lambert Drug Company on the subject. So Dr. Charles Scott approached him with the news that iproniazid could be the psychic energizer he was looking uh, for the reverse side effects of reserpine. Klein and colleague reported beneficial effects of iproniazid on a group of psychiatric patients in a meeting of the American Psychiatry Association. Their results were reproduced by many other research experiments. Hundreds of thousands of patients were treated off label. And they decided this is another paradigm shifter in the field of psychology, psychiatry. Now, what makes iproniazid act the way it acts is the question. The answer is in the biogenic mind and mood section of chapter 4. The author talks about biogenic amines, and when we say biogenic amines, we talk about serotonin, norepinephrine, dopamine, and their roles in neurotransmission. Norepinephrine utilizing neurons arise from the locus coeruleus in the midbrain, two nuclei on each side of the midbrain. These neurons are a few in comparison to other nerve cells, but they innervate most areas of the brain and spinal cord. These neurons regulate mood, pain, sleep, wake cycle. Serotonin uh, or 5-HT utilizing neurons, the neurons that use serotonin, also arise from the brainstem. Their nuclei are larger than the locus coeruleus. Biogenic amines are stored in vesicular structures at the terminals of these neurons. Action potentials trigger vesicular release into synaptic space, and please don't mind my anatomical, talking about the anatomical functions and physiological functions. So it's just because this helps understand better what we are talking about. And also, I assume that people who are watching this or want to watch this, want to read about it, are people with interest in chemistry, anatomy, physiology, etc., or in mental health or in general. So we were saying most biogenic amines uh, no, we were saying this action um, potentials trigger vesicular release into synaptic space. Then neurotransmitters activate cell receptors. Once the needed effect satisfied, the biogenic amines are removed, and this is within a positive negative feedback loop. Most biogenic amines are reuptaken and stored while others are metabolized. Protein molecules at nerve terminals are responsible for the reuptake. They are called biogenic amine reuptake molecules or transporters. New supplies of biogenic amines are resynthesized and pumped into the synaptic storage vesicle. The responsible pumping molecule is called VMAT, vesicular monoamine transport protein. Biogenic amines that are not properly stored are destroyed by the enzyme called monoamino oxidase. Now remember this because that's a very important enzyme for the field of psychiatry. Now this enzymatic activity was first described as tyramine oxidase by Mary O'Hare of Cambridge University when observed in the liver in 1928. Also remember tyramine oxidase. It was classified as a monoamino oxidase later by scientists who discovered large concentration of the enzyme in the brain. The enzyme lives on the outer membrane of the mitochondria, what we call the powerhouse of the cell. Biogenic amines, as was revealed, as we have seen, by 1950, are converted. Biogenic amines, as was revealed by 1950, by 1950, people have known this about them. They're converted to aldehyde by monoamino oxidase. That's why I said we have to pay attention to this enzyme. Then acids by aldehyde dehydrogenase, that is to say, norepinephrine, serotonin are converted to aldehydes by monoamino oxidase, then to 5-hydroxyindole acetic acid and 3,4-dihydroxymandelic acid, respectively. The action of monoamino oxidase and aldehyde dehydrogenase on dopamine gives off 3,4-dihydroxyphenolatic acid. The book says that COMT catechol or methyltransferase can methylate these byproducts to many other final byproducts. That is how the energizing effects of hyperoniazid understood in 1952. So out of all of this, you can discard all the chemistry jargon and just focus on one thing, that these enzymes are very important, and blocking them or potentiating them or enable them affects the brain working. Now, it was demonstrated that that is how the energizing effect was understood in 1952, right? It was demonstrated that is a potent monoamino oxidase inhibitors, and that's what I just talked about. Experiments on animals showed that iproniazid increased levels of norepinephrine and serotonin in the brain. A reserpine antipsychotic, the antipsychotic we have seen in chapter 3, blocks VMAT protein from transporting and storing biogenic amines in nerve terminal vesicles. Iproniazid, as monoamino oxidase inhibitor, prevents the degradation of biogenic amines and allows their storage at nerve terminal vesicles, as had Nathan Klein understood. Now let's talk about the rise of tricyclics. In 1957, depression acknowledged now as a diagnosis, treatment, and attempts to explain the disorder process and the mechanism of prescribed pharmacological action. Biogenic amines' roles in mood disorders have been accepted, and theories have been developed around them. Nathan Klein, American psychiatrist, has been awarded the Lasker Award 2 times in USA, and that's an equivalent of a Nobel Prize worldwide. Iproniazid has been the first monoamino oxidase inhibitor, and others were to follow. Many others were created and developed. They are now called antidepressants and no longer called psychic energizers. Side effects, unfortunately, hepatotoxicity. As a monoamino oxidase inhibitor, it can lead to fatal hypertensive crisis if the patient consumes food high in tyramine, such as cheese. And remember when I told you, remember tyramine oxidase? So if they eat lots of cheese with tyramine, they can that can lead to hypertensive crisis, and we'll understand why. Because this very problem caused the medication to be withdrawn back then. How and why it does so? Biogenic amines neurotransmission extends to the PNS, the peripheral nervous system. Norepinephrine is a biogenic amine that is a powerful vasoconstrictor. It is stored at nerve terminal vesicles. Tyramine is also an amine that gets stored in nerve terminal vesicles. Tyramine, although little and produced in the guts, it is metabolized by monoamino oxidase in guts and the liver. Now, as an amine, it is stored at nerve terminal vesicles. If monoamino oxidase is inhibited, it circulates in the blood, gets to the CNS, and as it does so, it displaces other amines from their storage, including norepinephrine. Vasoconstriction occurs, causing dangerous hypertension that can lead to infarction or death. Now we understand that uh, if we inhibit monoamino oxidase, this happens because we will have lots of tyramine circulating and getting to the nerve and getting stored also like a biogenic amines, like similar to biogenic amines because an amine also. And then as it gets to those terminals, it pushes away and displaces other amines, especially biogenic amines and norepinephrine. And norepinephrine is a strong vasoconstrictor. It constricts the blood vessels, and then that causes high hypertension, high levels of hypertension, and that can cause um, lots of problems to the body, infections, cerebral hemorrhage, you name it. Now, this can be controlled by restricting intake of food rich in tyramine, such as aged cheese, or just refrain from eating cheese altogether. In Chapter 3 of Drugged, the Science and Culture Behind Psychotropic Drugs, we have seen how phenothiazines were synthesized and how chlorpromazine came to fruition and how it was the first antipsychotic. Around the same time, Geeky Company, also in Basel, Switzerland, synthesized the phenothiazine chemically related structure to make a dye that is the research was suspended. They stopped the research. Ron Polonc was trying to find an antihistamine. So Geeky Company, it's written Geigy, company wanted to do the same, and they unearthed their immunodibenzyl, the one they synthesized earlier, and then synthesized many of its derivatives. It proved it has an antihistaminergic properties and other potentials. So they sent their immunodibenzyl analogs to clinicians to test on patients, and the analog they sent was G22150. Results were inconclusive when run by a psychiatrist that was named Roland Cohn when he used it as a hypnotic. So he asked for a similar product to CPZ. He said, I want something similar to CPZ or chlorpromazine. He was sent an immunodibenzyl compound with a dimethyl aminopropyl side chain that is structurally similar to CPZ. It was labeled G22355. It was imipramine, the first TCA that was birthed instead of an antihistamine. Now let's look at the structure. So this is chlorpromazine. You see these are three rings and this is the side chain. The difference, we have chlorpromazine and we have sulfur, and then we have this side chain here. The side chain is identical. Now look, this is a tricyclic. It has dose. There is no chloride. There is no sulfur, but you have the side chain is exactly the same one. So they sent them this product that was with a side chain similar to chlorpromazine and also the same similar structure, and it was the TCA. It did not work on psychotic patients, although it's like just a derivative of chlorpromazine, but still it did not work on psychotic patients. It improved symptoms in 3 patients with major depression when tested on them. Positive outcome obtained when tested on a larger patient population. Now, Dr. Roland Cohn reported these findings at World Congress Psychiatry in 1957 in Zurich. At the same time, Nathan Klein reported that iproniazid has psychic energizing properties. Now, imipramine, which is a tricyclic, uh, was marketed by Gihi Company as Tofanil in 1957. It received great enthusiasm in Europe and USA, and Merck, a big pharma, sold it in 1961 as amitriptyline or Elavil, the trade name. Many other TCAs were developed. Monoamino oxidase inhibitors with their deadly side effects faded off for TCAs. Now, TCAs are structurally similar to both typical and atypical antipsychotics, as we have seen, yet they act differently. Why do they act so, and what do they act on? TCAs and monoamino oxidase inhibitors have antidepressant effects. How? Monoamino oxidase inhibitors inhibit the enzyme monoamino oxidase. TSAs don't. They both obviously interact with the neurotransmission mediated by biogenic amines. That's what they have in common. They both interact with these biogenic amines, and when we say so, we talk about dopamine, we talk about serotonin, we talk about norepinephrine. The mechanism of tricyclic action, um, by 1932, it had been known that tissue could actively accumulate norepinephrine. And we know now, and we have known by then, that people can synthesize, and they synthesize norepinephrine, epinephrine, dopamine. So these synthesized biogenic amines, when irradiated, when turned radioactive by uh, mixing them with radioactive materials, uh, were they could be tracked accurately in the body. Radioactive epinephrine, norepinephrine injected in animals accumulated in peripheral tissue, while tritium used. Tritium is an isotope of hydrogen, while tritiated norepinephrine accumulated in dense sympathetic innervation sites, and then it's reuptaken by sympathetic nerve terminals. So they irradiated them, they tracked them in the body, and they know where they exactly know. They are sure where they are stored. Now they are sure how they get there. So the brain showed the accumulation of these in areas innervated by noradrenergic nerves. When they destroyed these, destruction of such nerves showed no accumulation. Synaptosomes. Synaptosomes are biochemical preparations of pinched nerve terminals. Those also prove accumulation of norepinephrine in terminals. Separate nerve terminal uptake systems were selectively expressed in nerve terminals. Their role is to terminate synaptic nerve transmission. So, what is the result of blocking such reuptake systems? Monoamine oxidase inhibitors, TSAs, tricyclic antidepressant, do so by blocking the reuptake of biogenic amines, TSAs, or by blocking their metabolism, such as the case of monoamine oxidase inhibitors, and then potentiating hence their synaptic action and prolonging it. 1960s, biological psychiatric theories emerged supporting the belief that depression and mood disorders result from chemical imbalances. We have seen actually this even before, but now it's like they're convinced now that this is what caused mood disorders. The proteins responsible for the reuptake are located in the presynaptic nerve terminal membranes and are known as norepinephrine transporters, dopamine transporters, and 5-HT transporter. Those protein transporters have many amino acid sequences shared. They also have shared functions and their overall structure, but have a substrate specificity. That is to say, they relatively selectively interact with a designated neurotransmitter. They also interact selectively with drugs that block or inhibit their metabolism. That is to say, for example, imipramine is more potent at inhibiting serotonin, while desperamine is more potent at inhibiting norepinephrine. TSAs have also come with their own lethal side effects, anticholinergic side effects related. So they cause dry mouth, constipation, and very, very worrisome arrhythmias. They also have been known, and that I read somewhere else, not in this book. It's hard to reverse an overdose when it happens. The search began for a new line of therapy with less lethality. It was found out in 1970s that serotonin has a role in the depression of, and serotonin theory emerged after autopsies uh, of depressed suicide victims revealed low levels of 5-HIAA byproducts. That is 5-hydroxy indolacetic acid in the brain and CSF. It's a main metabolite of serotonin. So low level of this means there has been low levels of serotonin. So they started searching for drugs that potentiate serotonin-mediated transmission. That is how Prozac happened. I'll talk to you about that in next time. My mouth has become dry, so I'll see you in next one. I'll see you in next one, and we'll talk about Prozac, about the phenomenon of Prozac, and about a few more things. And then I hope you enjoyed what I've shared with you. And then, as I told you in the introduction, if you read it, you can go ahead and read the book for your own on your own, and then post your comments or share your comments with me. Invite me to listen to you, to view your posts, and activate your bell. Subscribe, activate your bell, and then hear me out. So see you in the next one. Have a good night, everybody.